Hello students, in our last class we covered regarding the cell membrane of bacteria. We are dealing with the bacteria. Under the bacteria, the cell wall. Under the cell wall, we studied regarding the two types, that is the gram-positive cell wall and the gram-negative cell wall of the bacteria. And the terms gram-positive and the gram-negative, that we concluded by the gram staining process based on the behavior of the coloration which the bacteria absorb at the end of the process. That is the either it takes the pink coloration or the blue coloration. In case the bacterial cell wall which absorb the pink coloration, that is said to be the gram negative bacteria. In case bacterial cell wall appears in the blue coloration, that is said to be a gram positive bacteria. This is based on the cell wall presence, the composition of the cell wall presence. The gram negative bacteria which shows the pink coloration, the gram positive bacteria that shows the blue coloration. And again we seen regarding the layers of the cell wall, where under the gram positive there is only the one layer, while in the gram negative there is the presence of two layers of the cell walls. The thickness of cell wall is thick in the gram positive, whereas the thin in the gram negative, which measures 20 to 80 nanometer thickness in the gram positive bacteria, whereas in gram negative it measures 8 to 10 nanometer thickness. The outer membrane is absent in the case of the gram positive. The outer membrane is present in the case of the gram negative. Due to the presence of the outer membrane, the bacterial cell wall takes the pink coloration in the case of the gram negative bacteria. The chemical composition in gram positive seen is peptidoglycan, teichoic acid, lipoteichoic acid, whereas in gram negative lipopolysaccharide, lipoproteins and peptidoglycans. These are the chemical composition were seen in gram positive and gram negative respectively. Porins proteins are absent in gram positive, whereas the present in the gram negative. Lipid is less content in gram positive, recorded more in the gram negative. Peptidoglycan is more in gram positive, whereas less in gram negative. The permeability of molecules is seen more penetrable in the gram positive, where less penetrable in the gram negative cases. The resistance to the molecules is less in gram positive condition, whereas the resistance to the molecules more in the gram negative condition. Here in the gram negative we see the presence of outer and inner membrane, which is not seen in case of the gram positive bacterial cell wall. Today we will see the plasma membrane, the plasma membrane present in the bacteria. Before we go to the plasma membrane, we will recall regarding the plasma membrane, its function, its structure. We studied in our lower class regarding the fluid mosaic model, how the transportation of the ions takes place, whether it takes place by the active or by the passive transport, that is the active ions, how they transport in the cytoplasm through the plasma membrane, that we recall with the help of this video. Next we will see regarding the plasma membrane.
Membrane transport. So how do substances move into and out of a cell? Well, cells have a plasma membrane that acts like a fence, and this plasma membrane is what regulates what moves into and out of a cell. The membrane is comprised mainly of phospholipid molecules and has protein molecules scattered throughout its structure. The phospholipid molecules have a polar hydrophilic water-loving head and two nonpolar hydrophobic water-fearing tails. The membrane is formed as the heads face out and the tails face in, forming a bilayer, as you can see here. The inner part of the cell, called the cytosol, is here, and the outside of the cell is surrounded by a fluid called interstitial fluid. Substances move into and out of a cell through several different processes called membrane transport. There are two main processes: passive transport processes and active transport processes. The main difference between the two is that passive processes do not require energy expenditure, and active processes do require the cells to expend energy. Let's start by looking at the passive processes, which include simple diffusion, facilitated diffusion, and osmosis. Diffusion is the movement of a substance from where it has a high concentration to where it has a low concentration, or the tendency of a substance to spread out evenly over a given space. For instance, when a sugar cube is dissolved in water. Over time, the cube will dissolve and eventually spread out evenly in the water until it reaches equilibrium. Diffusion occurs down a concentration gradient, which is a difference in concentration of a substance between two areas. So the sugar molecules will move from an area of high concentration to an area of low concentration. Cellular diffusion is when diffusion of a solute, which is a dissolved substance, occurs across the plasma membrane from an area of high concentration to an area of low concentration. This is dependent on the concentration of a substance in the interstitial fluid outside of the cell and the cytosol inside of the cell. This can occur through simple diffusion or facilitated diffusion. Simple diffusion occurs with solutes that are small and nonpolar. By being nonpolar, they can move in between phospholipid molecules that form the plasma membrane because the interior region of the membrane is nonpolar. Some of the materials that move by simple diffusion include the gases oxygen and carbon dioxide and small fatty acids. So if there's a higher concentration of oxygen O2 molecules outside of a cell, they can move down the concentration gradient, across the membrane without assistance, and into the cell as long as the concentration gradient exists. And if there's a higher concentration of carbon dioxide CO2 molecules inside a cell, they can move across the membrane without assistance, out of the cell into the interstitial fluid. So again, simple diffusion is when nonpolar molecules pass directly through a membrane. The second type of diffusion is facilitated diffusion. This applies to solutes that are small and either charged or polar. Because these solutes are polar, the nonpolar phospholipid bilayer blocks them from passing through the membrane and into or out of the cell by simple diffusion. However, they can pass into and out of the cell with the assistance of plasma membrane proteins through a process called facilitated diffusion. There are two types of facilitated diffusion: channel-mediated diffusion and carrier-mediated diffusion. The difference between the two is the type of transport protein used to move the substance across the membrane. Channel-mediated diffusion is when an ion, which is a charged particle where its total number of electrons does not equal its total number of protons, giving it a positive or negative charge, moves across the membrane through a water-filled protein channel. Each protein channel is typically specific for one type of ion, and there are two types of channels: a leak channel, which is continuously open, and a gated channel, which only opens due to a stimulus and only stays open for a fraction of a second. So, for a sodium-positive ion, it can pass through a sodium-positive leak channel continuously, and a gated sodium-positive channel will only open due to a stimulus to allow the ion to pass through into the cell. Carrier-mediated diffusion involves the movement of polar molecules, such as simple sugars or simple carbohydrates, and amino acids across the membrane. This is accomplished by a carrier protein, which actually changes shape in the process. For instance, glucose binds to a carrier protein, which changes shape and moves the glucose molecule to the other side of the membrane. Now for osmosis. Osmosis is the passive movement of water through a selectively permeable membrane. This occurs when there's a difference in concentration of water on either side of the membrane. This can happen in one of two ways. Water can slip between the phospholipid molecules that make up the plasma membrane, or through integral protein water channels that are called aquaporins. The plasma membrane is not permeable to most solutes, such as charged, polar, and large substances. So, for example, one side of the membrane, the cytosol or interstitial fluid, can have more solutes than the other side. Let's say the interstitial fluid has 3% solutes and 97% water, and the cytosol side has 1% solutes and 99% water. In this example, water will move down its concentration gradient, from the 99% cytosol side to the 97% interstitial side to achieve equilibrium. Now let's look at active processes. Active processes require the use of cellular energy for membrane transport. There are two types of active processes: active transport and vesicular transport. Active transport is the movement of a solute against its concentration gradient, or going from an area of low concentration to a place of higher concentration. Vesicular transport is the transport of large substances across the plasma membrane by a vesicule, which is a membrane-bound sac filled with materials. Active transport has two types: primary active transport and secondary active transport. In primary active transport, cellular protein pumps, called ion pumps, move ions across the membrane against their concentration gradient. Let's take a look at an example. Here we have a membrane along with a sodium positive and potassium positive protein pump. 3 sodium ions and ATP bind to the pump, ATP being like fuel or the form of cellular energy. In order to release that energy, ATP breaks down into ADP and P. P binds to the pump, and the release of energy causes the pump to change shape and release the sodium positive ions into the interstitial fluid. 2 positive potassium ions bind to the pump, and the P produced by the ATP is released into the cytosol of the cell. The transport protein, or pump, reverts back to its original shape, releasing the positive potassium ions into the cytosol, and the pump is ready to do its job again. In secondary active transport, a substance is moved against its concentration gradient by using energy provided by the movement of a second substance down its concentration gradient. So, a substance that is moving from a place of high concentration to low concentration will provide the energy to move a second substance from a place of low concentration to high concentration. There are two types of secondary active transports: symport, where two substances are moved in the same direction, and antiport, where two substances are moved in opposite directions. In our example here, we have positive sodium ions moving from high concentration outside of the cell to lower concentration inside of the cell. A symporter will use the energy created by this to move a glucose molecule from a low concentration area to a high concentration area. And here we have an antiporter, again using the energy from moving positive sodium ions from high concentration outside of the cell to lower concentration inside of the cell, and H positive is moved up its gradient and outside of the cell. Vesicular transport involves the transport of larger substances, such as proteins or large carbohydrate polysaccharides, across the plasma membrane. There are two types of vesicular transport: exocytosis and endocytosis. In exocytosis, materials are secreted from the cell to the interstitial fluid outside of the cell. Typically, membranous vesicles, formed by the Golgi apparatus, transport materials to the cell's membrane. At the membrane, the phospholipid molecules that make up the vesicule fuse with the phospholipid molecules that make up the plasma membrane, and the contents of the vesicule are released outside of the cell. The vesicle membrane now becomes part of the plasma membrane. In endocytosis, the plasma membrane kind of traps the substance by folding inward. The lipid bilayer then fuses to form a vesicle surrounding the substance. There are three main forms of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. In phagocytosis, a large particle is engulfed by the newly formed vesicle, and this vesicle fuses with a lysosome, which is a membranous vesicle that contains digestive enzymes that break down the particle into its component molecules. Phagocytosis is also called cell eating. Pinocytosis, which is also referred to as cell drinking, is when the plasma membrane folds inward and engulfs droplets of interstitial fluid that contain dissolved solutes that can be used by the cell. Receptor-mediated endocytosis involves using receptors on the outside of the plasma membrane. These receptors bind with molecules in the interstitial fluid, and the membrane folds, enclosing the receptors and the bound molecules, to form a vesicle for transport within the cell. One last thing to note: both vesicular transport processes, endocytosis and exocytosis, require energy. And that be the basics on transport across a membrane.
With the help of video, we recall the things regarding the plasma membrane, that is the transportation, ion transportation, the plasma membrane structure, its composition. Now we will see one by one what all present in the plasma membrane. Here the plasma membrane of the bacterial cytoplasmic membrane, both the same, whether it is the plasma membrane or the bacterial cytoplasmic membrane, is composed of a phospholipid bilayer, where we seen in the video the presence of phospholipid bilayer, and thus has all of the general functions of a cell membrane, such as acting as a permeability barrier for most molecules and serving as the location for the transport of molecules in the cell. This is the main function of the plasma membrane, where the permeability barrier, that is the ions, the intake outtake of the ions, the location for the transport of molecules into the cell. Next, in addition to these functions, prokaryotic membranes also function in energy conservation as location about which a proton motive force is generated. This is proton motive force is generated when the condition applies when in the active case, active transport case, whereas in the passive the force is not required. Unlike eukaryotes, the bacterial membranes accept mycoplasma and methanotropes. They generally do not contain sterols. Again, then what are these sterols? These are the essential components of the membranes. Their functions include controlling membrane fluidity and permeability. These controls, that is the sterols, which control the essential components of the membranes. Their functions include controlling membrane fluidity and permeability. Instead of sterols, here is the presence of hapenoids, which performs equal function to the sterols. Many microbes do not contain structurally related compounds called the hapenoids, which likely fulfill the same function. Unlike eukaryotes, bacteria can have a wide variety of fatty acids within their membranes. Along with the typical saturated and unsaturated fatty acids, bacteria can contain fatty acids with additional methyl, hydroxy or even cyclic groups. Here, with saturated and unsaturated fatty acids, here additional presence of the methyl group or the hydroxyl group or the even cyclic groups. The relative portion of these fatty acids can be modulated by the bacterium to maintain the optimum fluidity of the membrane. This is the structure. The presence of cell membrane, the proteins, carbohydrate chain, lipid bilayer, the protein channel. Here the structure. This is the outside of the cell. In case this is the plasma membrane, if we see from this view, this is the outer view. This part is the outer view of the plasma membrane, and rest this part is inner view. This is the inner view. Here we see the cell membrane, that is the presence of bilayered plasma membrane which contains the head and tail. Again, we seen the regarding the concept of hydrophilic and hydrophobic, that is the water loving and water hating. The presence of proteins, the protein channel, the presence of the protein channel. These are the lipid bilayers, the presence of the carbohydrate chains. This is regarding the structure of plasma membrane. This is regarding the rough idea of plasma membrane. Here, as a phospholipid bilayer, the lipid portion of the outer membrane is impermeable to the charged molecules. However, channels are called porins. These channels are called porins, which are impermeable to the charged molecules. Presence in the outer membrane and that allow for passive transport of many ions, sugars and amino acid across the outer membrane. These allow the passive transport of many ions, sugars and amino acids across the outer membrane. These molecules are therefore present in the periplasm, the region between the cytoplasmic and the outer membrane. The region between cytoplasm and the outer membrane, that is called the periplasm. In case it is a cytoplasmic area, this is the outer membrane and this area is called the periplasm. That is between the region of the cytoplasmic area and the outer membrane. The periplasm contains the peptidoglycan layer. This periplasm consists peptidoglycan layer and many proteins responsible for substrate binding or hydrolysis and reception of extracellular signals. Here, this periplasm contains the peptidoglycan layer which contains the many proteins which are responsible for the functions of such proteins is substrate binding, which helps in the substrate binding, or in the case of the hydrolysis, or in the case of the reception of the extracellular signals. Here the signaling takes place in the cytoplasm itself, that is the chemical signaling. The periplasm is thought to exist in a gel-like state. This periplasm is assumed to be a gel-like state rather than a liquid state. This is due to the high concentration of proteins and peptidoglycan found within it. This is gel-like state, not a liquid one, because these have a high concentration of proteins and peptidoglycan found within it. Due to this case, this is assumed that it may be a gel-like state. Because of its location between the cytoplasmic and the outer membrane, signals received and substrate bound are available and transported across the cytoplasmic membrane using transport and signaling proteins embedded there. Due to its position, that is the presence between the cytoplasmic and the outer membrane, here it helps to the signals received inside and the substrate bound are available and transported across the cytoplasmic membrane using transport and the signaling proteins. Again, these signaling proteins and transport proteins helps to the transportation of the ions and the signaling messages. This is regarding the plasma membrane in the bacterial cell. In next class, we will see regarding the flagella, that is the flagellum and the pili. We will see in next class in detail regarding the flagellum and the pili. Thank you.